So hello everyone, my name is Trang and I'm from the Center for Bioinformatics in Saarbrücken, Germany. So the topic of our research is deregulation of epigenetic marks is associated with differential exon usage in developmental genes. Okay, so that's a quite long of a title. So I will break it down to parts, and the first part will be explaining the term differential exon usage, which is central to our study. So we all know that during alternative splicing, we have this change in exon, the presence or the absence of the exons would affect the inclusion of the exon. And so we measure that with exon usage, meaning that if a variant contains this exon 1, it will have a higher usage for this exon. And for the exons, other exons is the same. And what we look at specifically is the changes in exon usage, and we call that differential exon usage, or DEU for short, which basically happens when we compare two different cell types or cell type of different conditions together. Secondly, we have another important concept that is differential histomodifications, which carry the same idea that we just compare the histomodification enrichment in different cell types together. So from now I will be calling that DHM for the sake of simplicity. So we know that the local enrichment of histomodifications has been linked to different subtypes of exons in the population with transcript variants. And we are interested in modeling the local effect, the local enrichment of histomodifications on the splicing decision. And that actually is the first goal of our study. Then secondly, we know as well that alternative splicing and histomodification has been linked extensively to developmental processes because it creates a large reservoir of transcript and phenotypes and whatnot. So in the second part, we looked at whether the connection between the locally enriched histomark and the exon usage or the splicing decision would have something with developmental processes. And we achieved that goal by looking at 19 cells and tissues coming from the Human Epigenome Atlas. That is the part of Roadmap Epigenome program. So now we'll go more into detail what we have done on this data. So specifically, we use two different types of data. That is RNA-seq for the differential exon usage analysis, is DEU again. And we use chip-seq data for differential histomodification, DHM. And for that analysis, we performed it for 5 different histomarks. For both of the differential analysis, we perform pairwise comparisons. That means we compare the 19 cells and tissues all together in a pairwise manner. Of course, after that, we have to perform multiple testing corrections to take into account the blow-up error that come from all the comparisons that we have. But so after the correction, we'll be able to link the two features together, see how they are connected by Pearson correlation, and at the end of the workflow, we have something that we called in the study episplicings. So I will just go directly into details are the steps that we have done. So first of all, we define a flattened exon model, which will generally give us unique statistics for each exon regions if the transcripts are overlapped. So basically, if transcript variants are overlapping to each other, then the new overlapping or non-overlapping regions would define a new exons. Secondly, we define exon flank, which is the regions that we want to map or annotate the histomodification to. And those are the regions that's spanning 200 base pair up and downstream from the splice sites. So continuous to the pipeline, we have differential exon usage analysis and differential histomodification, and we perform those two with DSEQ and MA norm respectively. We filter out the DAU events that are significant with an FDR of less than 0.05. Meanwhile, for DHM, we also use the threshold for p-value and an m-value, which has the absolute value of more than 1. So naturally, now we would have a unique statistic for each exons and for 2 different types of differential features. And like I said before, we have DEU analysis, we have 5 different DHM analysis for the pairwise comparison between 19 cells and tissue. So in total, the number of comparison goes up to 1,000 or something. Of course, we have to take that into account, and that's why we perform multiple testing corrections, where we perform global analysis that would compare one cell type to all the other cell type. And we filter only the event, also the DEU and the DHM event that happen in both pairwise comparison and global comparisons. Secondly, we want to also look at the tissue specificity DEU. And that's why we filter out only the DEU that happen in less than 90% of the pairwise comparison. So it's demonstrated in this figure. Last but not least, we know as well that histone modifications is often linked to transcription. And we actually want to take that out of the picture because we want to focus on splicing only, right? That's why we eliminate all the first exon in all the transcript variants that are in our reference genome. So after the step of correcting our results, we went on and looked at the overlap between the 2 differential features as well as the correlation. But I don't think I will have enough time to talk about the co-occurrence, so we just focus on the Pearson correlation results. And like I said before, the result of our workflow is episplicings, well, that's what we call them. And they are defined by an R value, the coefficient of Pearson correlation that has absolute value of more than 0.5, which define a moderate to strong correlation. So on the left side, is 2 examples of episplicings that we found out, that is LMNB1, detected in the comparison between neuronal stem cell and pancreas for 3 different histone marks, and FGFR2 in the pair of mesenchymal stem cell and sigmoid colon for this histone mark A3K27M3. So we would focus more on this FGFR2 because it has an interesting story to it. That is, well, FGFR2 is a famous gene, and it has 2 transcript variants that are notably important in the transition, the epithelial and mesenchymal transitions. So it has 2 transcript variants, that is C and B variants. The C variant is most often found in mesenchymal cells and contain the exon number 23, while B variants would contain exon number 22 and are most mainly present in epithelial cells. But it's a little bit confusing here because we are using the flattened exon model. So the exon number we are referring to here is actually not the same as the one in reference genome. So anyhow, like I said before, we look at the exon usage in 2 different conditions. This is mesenchymal cells and sigmoid colon signified by 2 different lines of different colors, and then the enrichment of A3K27M3. From this exon usage analysis, we found that there's actually a DU event in exon number 22, which tell us that sigmoid colon actually contain more of the B variants and mesenchymal stem cell contain more of the C variants. So that actually checks out with the theory that we had before. So that's good. And then the result of our analysis pointed to the fact that, pointed the result that there's an anti-correlation between this histone mark A3K27M3 and exon usage. As you can see here, the red line is going up while the exon usage specifically just go down. And that is really interesting because we as well found experimental result that validate this finding that was just published, I think 2 years ago, that say that they've also found the anti-correlation between this histone mark here and the inclusion level of the exons in mesenchymal cell. And the histone mark that we found to be significant here is also linked to the deregulation of the other exon in the B variant. So the next step was to profile all the episplicings that we have gathered, and we did that by firstly look at the pairwise distance between the tissues based on the number or the amount of episplicings that they share. We did that by computing the shortcut index, And with that pairwise distance, we were able to construct, to perform hierarchical clustering. And the next step was to, well, we know that each cell and tissue would be related to each other in some way, right? Like they would have true biological annotations, like where they come from, the potency, and so on. And we wanted to know whether those true biological annotation would be matching to the clusters that we have based on the amount of similarity of the episplicings between tissues. And so we computed the adjusted Dren index to measure this matching degree. At the end of the line, we also look at the context, the biological context where episplicing potentially would be enriched in. And sorry, I kind of messed up there. Can you help me? Sorry. I know it worked now. Yeah, I kind of like. Oh, it's all right. Yeah, so we perform gene ontology enrichment analysis. Sorry for previous. Anyhow, I'm showing you now the results coming from the hierarchical clustering that we have. So here are shown here are the heat maps for the pairwise distance between tissues based on the shared episplicings. And for all the histomark that we investigated, we see clear cluster of tissues, cells that develop that belong mostly to the early stage development. That is, for example, stem cell, trophoblast, endoderm, ectoderm, mesoderm. And on the other side, we will have all the differentiated cell clustered together, signifying that they do not share that many episplicings altogether. And again, we want to look at whether this clustering would somehow match with the biological, the true annotation. And we specifically want to look at 4 aspects that describe a cell. So first of all, for example, potency we have, or the sample type of the cells, or origin or life stage of the cells. And again, we computed the adjusted Ren index to see the matching of the label. On the left side, again, we have this table showing you the value of the adjusted Ren index, and those are moderate or high. And so we conclude that episplicings actually have distinguished the cells based on the 4 criteria. One question that we often get from our reviewers or audience or readers is that, do we have to go that far and connect the 2 features, differential features together? Would that make difference? And why are we comparing cells to tissues? Well, I would say that this is here, what you are seeing here is the result from our supplementary study that show if we only take into account DEU or DHM, the clustering is not that clean and nice as we have seen in the previous slides. So something chaotic is going on here. And at the end of the workflow, we again perform gene ontology enrichment analysis for each of the set of episplicings that we gather for each histомodifications. And we group all the terms together in 3 broad categories. And the category that come out to have the most terms enriched is developmental processes. And also metabolism has also a lot of terms coming up. We pull all the episplicings together, regardless of which histомodifications it related to. We also see terms like morphogenesis or neurogenesis coming on top. So now it is already the conclusion that we found the differential incident between exon usage and histomark are often happening at the exon boundary region that we call flank, and many of the episplicings are differentially regulated at the exon level and are functionally enriched with developmental and metabolism processes. So we have a few outlooks to extend this line of work, and actually we have performed this study to look at the splicing factors that could potentially distinguish episplicings from non-episplicings, and it's actually the work from my colleague Hannah Robertson, which was presented in poster C302. Of course, now I cannot tell you to come and check out the poster, which is a shame. But you can connect to us on the live platform. And then we also want to look at the condition-specific episplicing genes or splicing factors that was, again, distinguish epispliced to non-epispliced genes. With our developed tool, PPI Express, and if you are interested in condition-specific protein network, then you could also check our tool. It is on live now, but we will have a paper announcing its presence to the world, I guess, soon. So that would bring me to the end of the presentations. Those are my colleagues who I appreciate so much for the support. That's Center for Bioinformatics, and we all, me and my group, we thank you so much for your attention, and I'll be getting questions for now.
Thank you very much for your talk. RNA-seq or gene expression data and chip-seq data are two omics modalities that have very different dynamic ranges and also noise levels, right? So let's say like the quality of the data is not comparable. So are you taking this into account when you are computing correlation between the two?
Yes, so we try to maintain the quality of the samples that we use. So first of all, we did a rigorous filtering of samples so that they would contain a lot of replicates, valid replicates. And with the algorithm, we try to correct in every step to reduce the error. Like, for example, for DAU, we only, at first, actually, we took a very low threshold for DAU analysis. And for DHM is a little bit higher. I have a more detailed number in the paper that we, have, but certainly we did multiple rounds of multiple testing corrections and filtering and adjustment of p-values, and yes, and that's why we have only 19 cells and tissues, because this compendium of data is vast. It's supposed to be the reference genome for human. But we only gathered 19 cells and tissues because we actually just really omit the data that do not meet the criteria at the beginning. So I hope that answers.
So actually, I have one more question. So you showed this example of K26 trimethyl being associated to this splicing of this one gene, right?
FGFR2, yes.
Yeah. So do you then find the opposite with K27 acetylation? Because I think you can't have acetylation and trimethylation on the same histone tail simultaneously. So you would expect that if one goes one way, then the other one should correlate the other way.
Actually, yes. I mean, this method allows us to see the genes expressed as an usage and histonification in different contexts, right? So like we perform 5 DHM analysis. So yes, sometimes we also see that there is like a combination of histones that one could be correlated and the other would be anti-correlated. Or we also see sometimes A3K4 MET3 would be correlated with acetylation, something like that. So it kind of confirmed that. But we should also be looking into this because maybe there's a motive, like a combinatory effects between the histone marks. That's interesting.
Even if there's no combinatorial effect, it might just be interesting to see that, trimethyl on K27 usually indicates, I think, closed chromatin.
Yes.
Acetylation indicates open chromatin. So they tell you the opposite story. It's just very strong evidence.
Yeah. Right? Yes. Yeah, we actually found some cases for that.
Great talk. So you told us that you took out the first exon because you wanted to exclude changes in expression because you're looking at splicing. But actually, I think that if you take this data now and you have it and try to look at it, being minded that some of the effect is going to be from the expression, it's going to be super interesting. to see how even chromatin in the promoter, not in the exon 1, actually affects splicing downstream or affects maybe histone modifications inside the gene. So that's also something that you can look at, see how one affects the other. So there's a growth there, I think.
Yes, I think the beauty of this workflow is that you can add more, or you can add more variables to it and make it scalable. Actually, you can have as many variable variants that you like. It show you some interesting idea, results.